thank you everybody for joining us today. My name is Catherine Warwick. I'm an applications manager with the UK and Ireland team for BMG LabTech. I'm very lucky to be joined today by Dr. Ben Allsop from the University College London, and we're going to be together talking to you guys about the use of microplate readers for simulating an ischemia-reperfusion type model, and how you can read multiplex different assays and test cardioprotective drugs in that environment. So I'm going to be passing over to Ben to actually show data on their project, but I'm going to take a few minutes beforehand to introduce the instrumentation that Ben has used in his project, and also, as we're here at SLAS launching our brand new CLARIOstar Plus, to talk about some of the new features that are available in that model on top of the existing CLARIOstar. So to start off with just talking about the CLARIOstar itself, it's a multi-mode reader that reads all non-isotopic read technologies as listed there. So Ben's going to be talking primarily today about fluorescence, luminescence. He's also utilized some time-resolved fluorescence fluorophores in his work as well. The CLARIOstar is an incredibly sensitive machine. It probably still is the most sensitive monochromator on the market. It uses linear variable filter monochromators. I'll come on to talk a bit more about that. But we use basically dedicated technologies, so we're using the right tool for the job in all situations. So UV vis spectrometer, filters where necessary, and a laser for alpha screen applications. So it's pretty flexible in terms of the assays it can take care of. The linear variable filter monochromator, patented to BMG, okay, it can read scan wavelengths 2D, 3D. It's got filter-like sensitivity. Most people don't use monochromators because they worry about sensitivity. With the CLARIOstar, you don't have that issue, you don't have that concern. Also, uniquely to this product, you can not only select your wavelength, but you can also uniquely define your bandwidth. So anywhere between 8 and 100 nanometers. And that 100 nanometer area is something that other monochromator devices can't achieve. There are some applications where filters are just necessary, where you really need to get high energy in. These are things like time-resolved fluorescence and TR fret, also fluorescence polarization and alpha screen. And I've mentioned before also the UV vis spectrometer. Here we can capture full spectra in under 1 second per well. So this is a technology we employ across all of the instruments within the BMG range. So for those of you that may have also an interest in enzymatics, or for your cell-based assays for adding components to see how that changes their cellular response, we can add two things to the cells. So we have two injectors that can apply varied volumes across the plate in as little as 1 microliter per well. We also can do Z height adjustment from both the top of the well, but more importantly for cell-based assays, the bottom of the well. Right? Not a lot of plate readers actually offer this functionality, and it really makes a difference to getting the best sensitivity performance from your plate reader. Now, our fastest approaches with cell-based assays in mind, we know quite often they're non-homogeneous. They never grow in the places that we want them to grow. We have different ways of interrogating the data. Our fastest ways are through orbital averaging and spiral averaging. And we also have a way to create a color bitmap over the well by throwing like a matrix there. So we can actually get better reproducibility from well to well in that situation. Now let's talk a bit more about the gas control, because this is what's been really enabling for Ben and his work. Just to talk about it simply, it's independent control of oxygen and CO2 at the same time. So you can create any kind of physiological environment you want between 0.1 and 20% on both gas levels. All right, you can change the gas levels through the software, you can change it through the display panel, as you can see up there on the screen. And we did a talk yesterday with Promega as well. There are so many assays now that are tackling real-time assay scenarios. You get more information from the same plate, and literally proliferation, ROS, viability, migration. There's so many things now that can be done in a microplate in real time. So this is very enabling there. Now, the work that first got Ben interested in us was work we did as part of a Horizon 2020 grant that we call MetaCell, trying to find a full metabolomics solution in a plate reader. We've worked in conjunction with Incardia with their cardiac myocytes. We've worked with Agilent on their oxygen and pH sensor probes, and we had academic partners both at Oxford University and Imperial. And we developed technology to not only drop the gas levels or have that controlled to low levels, but also to be able to rapidly perfuse that environment. So this meant that studying stroke, cardiac arrest, cancer cycles for hypoxia and tumor progression could be studied within the reader. So users have complete control over that. And it's working as part of that project that our initial data in an application note we produced, working with the iPSC cells from Incardia, that we were able to show that we were able to multiplex multiple assays in one go. We were not only able to measure the oxygen in real time with our system, we were able to use the oxygen sensors to tell what the oxygen level really was in the cells and that the gas controller was tracking correctly. But we were also able to measure mitochondrial membrane potential and ROS from the same cell set. So it was very enabling from that perspective. So that covers, if you like, all the features that Ben has had access to up to now. I'm just going to finish on just highlighting some of the new points for the CLARIOstar Plus. We've really built on the CLARIOstar platform, so everything the CLARIOstar has, the CLARIOstar Plus has, but it has some extras as well. So the first thing to talk about is we've added enhanced dynamic range to this instrument. This gives a much broader dynamic range than any other competing reader on the market. Okay, you can go from femto to micro concentrations in one reading, but also we've designed this feature that no optimization steps are required at all. No focusing, no sensitivity optimization. In one go, you can read this full range and literally somebody put the plate in and press go. So for fluorescence and luminescence assays, this is highly enabling, especially when you're doing assay development. We see this instrument sit alongside our FerroStar quite often for both assay development, also for enzymatic studies. Here's an example on the left of an enzymatic study. Number one, traditional systems and even our systems can sometimes saturate if the wrong setting is selected. With enhanced dynamic range, there's no risk of missing out on your signal. It can just keep growing. But more importantly, actually, as an app scientist, we hear complaints sometimes that you don't see good differentiation across the whole kinetic curve. And because we're choosing the right sensitivity settings every step, according to every well and every signal read, you get better signal differentiation even at the low end compared to the top. So that works really well in that instance. We've also added in a red detector into this instrument. So far, we've always used low noise PMTs in the CLARIOstar, and the addition of this red shifted PMT is giving our customers a lot more choice. You know, people want to move to red for autofluorescence issues with cells. They want to get away from compound interference issues. So this is just giving our users a lot more choice. And as a consequence of putting this red detector in, we've actually found that even things that were in our existing range, things like Alexa 647, we actually can significantly improve limits of detection for those assays as well with the addition of this option. So we're still using our low noise PMT for luminescence and alpha screen, but now you have access to a fuller range of assays for the CLARIOstar. So, okay. Looking to the FerroStar, lots of people like the sensitivity that gives, and luminescence performance is fantastic there. We wanted to bring some of the features of the FerroStar into the CLARIOstar. As I said, it works often alongside the FerroStar as an assay development tool. So we've brought in advanced crosstalk reduction features. Crosstalk can happen in a glow assay, obviously from above the well, but not so many people realize that through the sides of your white plate, you can also be seeing light contamination. You see here, this is actually a white plate with a torch behind. You know, even an opaque plate will let light through. And actually what we're able to do with our algorithms is turn this kind of crosstalk into this kind of level just by using our system. So again, available for the FerroStar and now also for the CLARIOstar Plus. For Ben and users that utilize the ACU, we've introduced a new graphical interface. This just makes the multiplexing that I showed you already just far easier to to manage and actually to get going. You're actually able to visualize the cycles of gas ramping that you want to introduce, and actually able to export and import settings very easily with this system. So it just makes life a lot easier for users. So on that point, I'm going to summarize and pass over to Ben. But really with the CLARIOstar Plus, you've got all this flexibility. You're bringing your plate reader out of your hypoxia chamber, out of your incubator. You free up that space for something else, right? Do your microscopy in there or something else. We're also, the flexible tools that are available, the sensitivity and performance of the reader really accommodate a wide range of assays. And with the new CLARIOstar feature as well, I mean, it's all about hassle-free detection. You know, no optimization steps, broader dynamic range. And of course, for the gas control users, an easier way of working under more physiological conditions. So with that in mind, I'll pass over to Ben at this point. He's going to talk more about the data they've created.
Okay, thank you very much, Kath, and thank you for your time. So I'll be talking about an ischemia-reperfusion assay that we have been developing and the application of that assay for drug screening. So I don't work for BMG, I'm not being paid by them. I work for UCL in the TRO. The purpose of the TRO is to nurture any ideas and expertise at UCL and then try and bring them to the wider world. And I work in the Drug Discovery Group. And what we do is we take the best academic science and turn it into drug discovery projects. So we're medicinal chemists and biologists, focused on small molecule drug discovery, and we have to do a degree of target validation, assay establishment, and then we run biochemical and cell-based screens. We also manage collaborations between UCL and industry partners, and that's where our association with BMG has come from. One area of expertise at UCL is coronary heart disease, particularly the STEMI heart attack. A heart attack or a STEMI heart attack occurs when you get an occlusion in an artery and arteriole in the heart, and that prevents any oxygenated blood from getting to a region of the heart. So that gives you a period of ischemia where you're not getting any oxygenated blood in, and then when you break open that atherosclerosis or plaque, you then get reperfusion to that part of the heart, and these both come with their own associated injuries, which I'll go into in more detail later. But a STEMI heart attack is a major form of coronary heart disease, and it's one of the biggest killers in the world for the over 60s. Just for some context, in the US there are 550,000 new STEMI heart attacks every year, and then you get 200,000 recurrent incidents. So there's a problem not only with a risk of death on your first heart attack, but after you've had one, you're more likely to have multiple. The good news is that there is a decreasing incidence in Western countries, but that's mainly because of people becoming more aware of their cardiac health, stopping smoking, improving their diet. But as more of the world is becoming industrialized, places like China, India, Indonesia, you're getting a lot more obesity. A lot of those places still have quite high smoking rates, and you're getting more heart attacks. So it is a big problem still, and it is only getting bigger. So this project is a collaboration between UCL academics and the Drug Discovery Group, of which I'm a part, and has been supported by BMG initially through the loaning of CLARIOstar and ACU, and which we've now gone to purchase, and has also been supported by other academic and industry partners. So ischemia-reperfusion injury. As I mentioned, the ischemic and the reperfusive phase of a heart attack both have their own distinct injuries, and those contribute to an overall infarction. An infarction just means the degree of heart tissue that's lost when you have the heart attack. So the ischemic phase comes with acute ischemic injury. That's characterized by a reduced pH, a reduction in glucose utilization, and a movement to anaerobic metabolism. You get an accumulation of inflammatory markers as cells begin dying from necrosis, and you get imbalance in sodium and calcium. And then when you reperfuse with either thrombolytics or by physically breaking apart a plaque, you get a separate injury termed ischemia-reperfusion injury, and that's what the focus of my talk will be on today. Ischemia-reperfusion injury occurs because of the mitochondrial health deteriorating, and you this occurs mainly through the mitochondrial permeability transition pore opening. That means that the mitochondria can't regulate itself properly and doesn't function as it should. You also get calcium imbalance and the generation of reactive oxygen species. So together, these two separate injuries contribute to your infarction. Just as a side note, this is a focus on heart attacks, but IRI also plays a role in stroke and has a role in cancer biology. So Infarct size is extremely important. There's a meta-analysis from 2016 demonstrating that people who have a greater infarct size have a much bigger chance of death and rehospitalization within a year, which is demonstrated here. So you really want to reduce the infarct size as much as possible. And to highlight that, they also have a figure in this paper where people who have over a 30% infarction, so have lost over 30% of the heart tissue, are 7 times as likely to die or be rehospitalized within a year compared to the lowest quartile of people. And in people with the average infarction quartile, they're 3 times as likely to die or be rehospitalized within a year compared to that lowest quartile as well. The best way to reduce infarction size is to reperfuse the heart. The ischemic injury will cause a huge degree of infarction, up to 70% of the heart tissue if you don't reperfuse. So when you get to hospital, you're given a thrombolytic or you're given percutaneous coronary intervention, which is just an angioplasty to physically open up the artery. But when you do reperfuse and give glucose and oxygen-rich blood back, you do cause a paradoxical injury called ischemia-reperfusion injury. And that can mediate a damage of up to 25% of the heart tissue. So that's still a large infarct, and that is targetable and easy to modulate. Well, not easy, but we'll give it a go. So IRI represents an unmet medical need and a viable target to improve STEMI patient outcome. So the purpose of our project is to generate small molecules that can ameliorate IRI and can be given prior to thrombolytics. The good news is there are already multiple ways of ameliorating IRI reported in the literature. These are mediated through GPCRs, such as adrenomyelin, bradykinin, urocortin, and these are the basis of ischemic pre- and post-conditioning techniques which have been reported on, and there's a lot of data coming out of UCL on that. But as well as downstream from insulin and growth factors like IGF-1 and from cytokines like interleukin alpha. And what these all have in common is that by hook or by crook, they prevent the opening of the mitochondrial permeability transition pore. So we've used insulin to validate the assays that we've developed. And that's because insulin has got literature demonstrating that it protects against cell loss in isolated cardiomyocytes in vitro models, as well as the Langendorf mouse model, which I'll introduce in a bit. The data from that is here. And that mechanism of protection being via the mitochondrial permeability transition pore is also present in the literature. There's a lot of insulin, so you might be wondering why we don't just use that in clinical trials instead. So GIC treatment, glucose, insulin, potassium treatment, has been tried in clinical trials where it was unsuccessful. You give the glucose and the potassium to avoid a hypoglycemic shock, just as an aside. However, the post-trial commentary of this trial has been highly critical. The trial was cut short and the primary endpoint was changed so that it was the movement to infarction, which, considering that an ischemic phase mediates a degree of that infarction anyway, and that's not what you're targeting with insulin, was an extremely difficult barrier to overcome. But once you actually get into the data reported, patients who received the GIC treatment did have a significantly reduced infarct size and significantly reduced rehospitalization rates. They had reduced mortality, but that was non-significant. But I still think this data is very encouraging. So this is a schematic demonstrating the current models for in vitro modeling of IRI at present. So you guys may have seen this before. This is a hypoxic chamber. You put a flask of cells or a 6 or 24 well plate of cells in here and then attach nitrogen, flush out any oxygen, and create your hypoxic environment. You'd then place that in an incubator. Depending on the cell type, you will then leave it for 1 to 20 hours in that ischemic environment. So some cells, you only need 1 hour to induce a hypoxic insult that you can then generate ischemia-reperfusion injury. Other cells will require very, very long extended periods of time. And you can't really see what's going on during that time with this model. It was also determinant on whether or not you've got endothelial cells, stuff like that there. So then you'll reperfuse. You change from an ischemic buffer that we put on to just a normal buffer. And then you leave the cells for another 4 hours. And again, in this model, you can't see what's going on in those 4 hours. 4 hours is an arbitrary time point. Quite a lot of papers have also gone and seen what's happening at different time points after that. But it's commonly used in 4 hours in this instance, where you just have to then go for one endpoint measure. And they're quite often measured by propylium iodide and FACS. So this assay is a limited throughput. Quite often you only have 6 or 24 wells in here because you've got this as an output. And it's information poor. You don't know what's going on during this period. Another assay that I'll be referring to, and you have to forgive this video for not working, is the Langendorf preparation. This is a gold standard assay and really helps you model infarct size extremely well. So you remove the heart from a mouse or a rat, you cannulate and perfuse it, and then it will begin beating, and then you can then control that perfusion to induce a ischemic insult, and then reperfuse, treat with any drugs that you want to do. So this gives you measures of heart contractility, beat rate, and overall infarct size. So it's a really gold standard assay. It does have its own problems though. Firstly, you've got limited throughput. You have high animal usage, and it requires a skilled surgeon to perform. And it's also very expensive as a result, which makes everyone nervous. So we needed a new assay. So we approached BMG to use the CLARIOstar. We did this for multiple reasons. We knew we needed it to be in a plate reader so we could get kinetic data. But the addition of the ACU giving us quick ramping of oxygen whilst controlling CO2 and temperature was very valuable. This has allowed us to move into 96, and we're working on 384 well assays. And obviously it gives you real-time kinetics and the opportunity to multiplex, which is important. So the first thing we did was compare the CLARIOstar model to the hypoxic chamber model. And we did this by using these optical oxygen sensor spots to see the percentage of oxygen in the media in the hypoxic chamber, and compared what was going on with the CLARIOstar model using an intracellular oxygen diacetyl biadulant, which lets you see the percent oxygenation as you go through the ischemic insult. So we apply hypoxia here, and it takes almost 2 hours to reach 1%. And when we apply the hypoxia here in the CLARIOstar model, it takes 45 minutes to get down to 0.1%. This is slightly unfair as a comparison because here you can't have any cells present, so you're not getting oxygen used up as quickly, and you have a higher starting point. And if you did, you wouldn't be able to see what was going on in the cells. And there's more media in here as well because it's a 6-well plate. But what I think that highlights is if you could see what was going on in the cells, it would be in a plate reader, and we wouldn't have had to make this assay. So I still think that this is a very good point to be reaching. Another advantage was that our assay optimization was quite quick. So I mentioned before that you can have between 1 and 20 hours ischemia. When you are doing that in the hypoxic chamber model, that means you have to have a hypoxic chamber for every time point you want to test, and then never mind what you want to do for different time points on the reperfusion. Whereas here, what we can do is we can induce the hypoxia, leave the plate for 18 hours, we've done here, and then reperfuse. And what we're looking for is we're looking for the point at which cells start dying. So we've used CellTox green here. And in these cells, the H9C2 rat myoblasts that we've used in 96 well, they start dying quite quickly. And what we want is to identify when they start dying, go just before that, so that any cell death we then see on reperfusion is our hypothesis-driven toxicity, so the ischemia-reperfusion-driven toxicity. So we took 1 hour time point from this, and then were able to do a cell titration on the 1 hour time point. So this red line here is the set point chamber oxygen for the CLARIOstar. So that's what I've set the CLARIOstar to do. And that's consistent throughout all the further figures that you'll see in this presentation. So as you can see, we've got very little cell death going on during the ischemic phase. Down here, all the different cell densities are right at the bottom. And then on reperfusion, you get an initial bit of reperfusive shock. So you get a small bit of death. And then after a couple of hours, you start seeing this rock up. And this is the IRI mediated, largely apoptotic death, or we think it's apoptotic. This is why it's important to be able to look kinetically. So we then needed to validate our model, and we did that using insulin. So the first thing we wanted to do is characterize our insult. So on every plate that we run, we use this intracellular oxygen dye to just characterize and demonstrate that those cells have been through the insult and that the insult is uniform. So that our control and insulin-treated cells all went through the same insult. And then we were able to demonstrate that the insulin is also protective in this model. So again, we have little to no cell death occurring in ischemia. And then on reperfusion, you get a lower kick up with the insulin treatment. And then you also get a lower endpoint. So you're getting less of that IRI-mediated death, which is consistent with the published results. We also use 100 micromolar tretinoin as a positive control. But then once we've gone through that protocol, we have a plate that we can use for endpoint assays. So we typically use cell titer glow to report on cell death so that we can easily put that into our data handling software. But we can also investigate some of the more mechanistic modes of toxicity, for example, using DHE to detect ROS and demonstrate that the insulin treatment is reducing the amount of ROS that's generated. You also have a plate that you can fix, stain, and use for fluorescence microscopy. So here we've used mitotracker red dye, which will accumulate in healthy cells and will give a bigger signal when you've got more healthy cells. So during the IRI treatment, you get a reduction in the accumulation of the mitotracker red dye, and you also get quite obvious cell loss. And then with the insulin treatment, you have a lower degree of cell loss, and you have a rescue in the amount of mitotracker red dye accumulating in those cells. So this is quite nice for basic biologists. So we've then applied this to drug screening and stratifying our compounds. So we can treat with our compounds, see when they're causing their rescue, how they are rescuing, and that can inform our chemists on what they want to then do as they follow the SAR. We also can multiplex and look at ROS as an endpoint assay. And we are getting a degree of rescue in this model. Again, we also have used it with the mitotracker red and demonstrated a degree of protection and been able to look at the cells by fluorescence microscopy. So we envisage this model being used as a hit-to-lead or lead optimization model. It has advantages in that it allows you to test a disease-relevant and an in vitro model and a hypothesis-driven toxicity, as well as multiplexing multiple outputs for each experiment. I'm not sure quite how well this translates to an American audience, but in the UK we have the NC3Rs, which is an awarding of money to experiments or programs that are going to improve animal models. So we feel this ties in quite nicely with that, in that you have a reduction of the number of animals that you need to perform the Langendorf experiment on, because you can triage your compounds and reduce the size and cost of animal studies. Those are slightly more efficient to perform, and we're working to use human iPSCs in this model so that eventually it will become a more relevant model, or we hope it will become a more relevant model than the Langendorf preparation will be for human studies. So to conclude, we've developed a novel medium throughput assay, 96-well based, for basic research that also, I believe, has an application for drug discovery. Because it's a plate reader based assay, it's information rich, you get all of that kinetic data, and then you can characterize your insult by looking at internal oxygenation and, well, whatever else you're interested in. And you also have a plate which is suitable for endpoint assays, which can quite often be easier to get into data handling software. And we've recently started just using the CLARIOstar to do the insult on bigger plates, so 24-well plates, and then harvesting the lysates and looking at Western blotting, so we can stop it at different points, take the lysate out, and then start looking at things that we're interested in basic biology. And we believe this is useful to stratify hit-to-lead compounds prior to the Langendorf preparation and reduce the size and cost of disease modeling in animals. So that just leaves me to thank the TRO DGG, particularly Richard and Trevor, as well as the chemists, our collaborators at the UCL Hatter Institute, particularly Sean Davison and Derek Yellen, as well as BMG LabTech for inviting me to speak today, and the support throughout the project, and in particular Catherine for her support. And thank you. If you have any questions, I'd be happy to answer.